Welcome to the second part of the chapter Biomolecules. And in this class, I'm going to discuss about the carbohydrates. The term carbohydrates is very familiar to you. From class 1 or 2, we have been studying about carbohydrates, that they are the energy-rich food. The major sources of carbohydrates are, say, rice, wheat, the potato, etc. And they are the major energy giving food. Then in class 10 we studied about the digestion of carbohydrates. Now here we will discuss what actually these carbohydrates are. The term carbohydrates simply means the hydrates of carbon, where it was assumed that the carbon and water are present in a specific ratio. Like if I say carbon is X in number, then the water is present in Y in number, where hydrogen and oxygen are always in the ratio 2:1. But if you see the structure of acetic acid, that is CH3COOH. Here the carbon is 2, hydrogen is 4 and oxygen is 2. It can be represented as C2H2O whole twice. This fits into the definition of the hydrates of carbon. But we know acetic acid is not a carbohydrate. So, though the name says it is hydrates of carbon, not all the carbohydrates are hydrates of carbon. So, the recent definition of carbohydrates are, they are actually polyhydroxyaldehydes or polyhydroxyketones of organic origin, which can liberate energy on oxidation. So, these glucose, fructose, starch, if you analyze the structure of them, they are all polyhydroxyaldehyde or polyhydroxyketones. Now, these carbohydrates are also called saccharides, meaning sugars, because they are sweet in taste. Many of the carbohydrates are sweet in taste, so called the sugars. Carbohydrates are actually either aldoses or ketoses. If they have a terminal aldehyde group, they are called aldoses, and if they have a subterminal ketone group, they are called ketoses. The saccharides are of various types. The simplest of the saccharides are monosaccharides, which consists of a single sugar unit. The monosaccharides are again of different types. Like the simplest monosaccharides which are present in the body of living organisms are actually 3 carbon compound. For example, dihydroxyacetone or glyceraldehyde. Then there are 4 carbon containing monosaccharides called the tetroses. Erythrose is a tetrose. 5 carbon containing monosaccharides are called pentoses or pentose sugar. Heard about the ribose sugar, which is a part of DNA, deoxyribonucleic acid, or ribonucleic acid, that is RNA. The sugar present in DNA or RNA is a 5-carbon monosaccharide called ribose. Then we have the hexoses, the 6-carbon containing sugars. The most common one being the glucose, fructose, galactose. They are the examples of hexoses. The glucose is commonly called blood sugar, fructose is commonly called fruit sugar, and galactose is mainly called milk sugar. Then we have heptoses containing 7-carbon sugar. The next group of saccharides are the disaccharides. The disaccharides are formed of 2 sugar units joined by the glycosidic bond. Now, the examples of disaccharides are, say, sucrose, which is composed of 1 glucose unit and 1 fructose unit. The mannose, which is composed, or maltose, which is composed of a two glucose units, you can say. And then you have lactose, which consists of 1 molecule of glucose and the other molecule of galactose. Now, these simple sugars, many of them act as the reducing agent. And that is why their detection is based on their ability to reduce different substances or chemicals. The Benedict solution, the Fehling solution, which are used to detect the presence of glucose in urine, is actually based on this reducing property of glucose. They act as reducing agent and they reduce the cupric ion to the cuprous forms, thereby causing a color change which can be detected visually. Now, when small number of, say, 1 to 7 sugar units are joined to each other by glycosidic bond, they are known as oligosaccharides. Oligo means small, short. So, oligosaccharides are short chain of monosaccharides. And another group of carbohydrates which is very much prominent in human and the plant and the animal body are the polysaccharides. They are the polymers of monosaccharide. The most common one you have seen is starch, which is the storage form of the glucose in plants. It is a homopolymer of glucose, also called glucosan. That means many units of glucose are joined to each other to form the starch molecule. Now, glucose in the starch molecule are joined in 2 major ways, forming 2 parts of the starch. One is known as the amylose and the other is known as the amylopectin. The amylose form of the starch or amylose component of the starch can hold iodine molecule in its conformation and hence gives a blue-black color. And that is the test for glucose. Remember in class 10 we studied about the glucose, test of glucose or photosynthesis as the presence of starch with the help of iodine test giving blue-black color. That is because the glucose that was formed in the plant body was immediately converted into starch. The starch has 2 components, amylose and amylopectins. The amylose form can hold the iodine molecule in its 3D conformation and gives blue-black color. The next type of polysaccharide is your inulin, which is a homopolymer of fructose. Glycogen, which is stored in human body, is the storage form of glucose and is also a homopolymer of glucose. Similarly, chitin in fungus, the cellulose, they are all polysaccharides. Now, carbohydrates have got immense function in our body. They are the major energy giving food of our body. Oxidation of carbohydrates release huge amount of energy stored in the bones, or huge amount of energy by formation of ATP. And when ATP breaks, it releases huge amount of energy.